Good morning, everyone. My name is Diana Wall. We're going to fix this in a second. My name is Diana Wall, and I'm the science chair for the Global Soil Biodiversity Initiative that puts on these wonderful webinars. And I want to thank all our panelists ahead of time and Katie Field, our moderator. I just have a couple of housekeeping rules and then I'll go away. One, you put your questions in the Q&A, but only Katie will be looking at those, and then she will send them to the panelists. We will not answer those. The panelists will not answer those individually. They will all go to be on as part of the discussion. The other thing is we're recording this. And so if you have any problems with that, you need to go away. Because what we do is we take all our webinars, our past webinars, they're all on YouTube, and people use them from everything from class, for seminars, and refer back to them as they're studying a particular subject. So with that, I know you've already recognized the panelists, but I'm going to turn it over to Katie, who is going to be your moderator today.
Great. Thanks, Diana. I think that's my cue to get started. So I'm just going to share, I've got some introduction slides. It's a pleasure to be here today and to be talking to you all about this very exciting and topical topic on threats to mycorrhizas. So I'm just sharing my screen. Hopefully you can see this. So what I'm going to be doing now is just going through a bit of a broad background about why we should care about mycorrhizas in the first place. I know I'm speaking to the right audience here. And then I'll give you a brief introduction to our panelists this afternoon, this morning, tonight, wherever you may be. So, threats to mycorrhizas. Now, we often get told that Earth is a blue planet. That's because the majority of its surfaces are covered in oceans. However, that doesn't represent how important the most important organisms on Earth, which in my opinion and the opinion of many others are plants. And so we're probably better calling it a green planet rather than a blue planet. Now, luckily for me, the BBC was listening to my call when I was telling them it shouldn't be called the blue planet anymore. And they did actually take up this and made it into a series, which is excellent. So if you get a chance to watch that, I recommend watching it. Anyway, plants are really important and they make up a huge proportion of Earth's terrestrial biosphere. Almost all of the carbon on Earth is a result of plants or is plant derived. But it wasn't always like this. If we go back 500 million years, there were no plants on Earth's land surfaces, and it was devoid of any sort of terrestrial visible life. Certainly there was microbes and fungi, but no plants. But fast forward about another 100 million years before that, so around 400 million years, and Earth's landscapes might have looked a bit like this. And you have encroachment of early land plants, simple branching structures, but in the background, you can see huge towering fungal structures called prototaxites. Now, obviously, this is an artist's impression. We don't have time machines, so we don't know exactly what it looked like 400 million years ago. So how do we know what how do we know what these landscapes look like? I mean, this is Freie, which is a picture. This is a small village in Aberdeenshire in the UK, which that recreated landscape was a depiction of. And this is it today, and there's nothing to see there. It's just it's green farming land where sheep graze. However, if you scratch the surface of Freie, you uncover these incredible fossils. And it's these fossils that give us real clues as to how that plant life came onto land in the first place. So because of the unique environmental conditions at Freie during that time, the preserving power of the water was incredible. And it was so great that it was able to preserve organic matter almost entirely intact. So this is a microscopic section, transverse section, a slice through these axes of this plant Aglaophyton major. And the preservation is so wonderful. You can make out individual cells of that plant. You'll also notice this dark smudgy ring on the inner cortex, just running around here. If you zoom in on that section, you can see structures that look like this. Now, I know many of my my panelists will be looking at these going like, ah, I know these. And that's because they look almost identical to modern day mycorrhizal fungal structures that are formed inside the roots of nearly all plants today. So these are arbuscular mycorrhizas, and they're the ones that I'm particularly interested in, but we're going to talk about a load of different types of mycorrhizas today. But you can see just by comparing these structures formed in a root of a wheat plant here with these 400 million year old mycorrhizal structures, and it gives us clues that these fungi were probably important in helping plants get onto land. Why, I hear you ask, how might these fungi have helped these plants? Well, it's all to do with what mycorrhizas do. So today, nearly all plants form mycorrhizas, and their primary function, or one of the main functions that we study, is their capacity to increase plant access to mineral nutrients in the soil. What they do is they assimilate these nutrients from beyond the root depletion zone or from sources or pockets of pores in the soil that the plant roots can't get to, and they assimilate those nutrients and transfer them to the plant they're associated with. In return, the fungus takes carbon compounds such as fats and sugars and transfers them to the fungus living in the roots. So you have this bidirectional exchange of carbon for nutrients, which is characteristic of all mycorrhizal symbioses. So we normally think of it as being this mutualism, but in reality, it lies anywhere on this scale, from being completely mutualistic, where everyone benefits, to being parasitic, such as in this example here, where this plant has lost all of its chlorophyll and relies entirely on the fungus that's connected to another plant to get all of its nutrients and minerals that it needs. You also have plants that live life somewhere in between, and they switch or oscillate between being parasitic or mutualistic or anywhere in between there at any given point in time. And lots of different factors can affect where a particular mycorrhizal symbiosis lies on this continuum. And that includes things like competition, genetic diversity, but most importantly for today, things like soil nutrient status, soil type and disturbance, or atmospheric carbon dioxide can play a role. Now, we've done loads and loads of work in wild plants as a community, and we've found out that these mycorrhizal fungal associations they can increase plant access to soil nutrients, and they can enhance plant defenses against pests and diseases, and they've also known to increase drought tolerance and resistance in their host plants. However, they're not invincible. They face many threats, and that's what we're going to explore in our discussion today. So we're going to be exploring conservation and preservation of mycorrhizal fungal diversity, how we can make those critical unknowns known, and also the influence of global change. And that's thanks to today's speakers. So I'm going to stop sharing my screen, and I'm going to just briefly introduce our speakers. If you'd like to turn their cameras on, I shall hand over to them in just a moment. So we've got Matthias Rillig from the Freie Universität in Berlin. We have Nguyen from the University of Hawaiʻi in the US. And we have César Marín from Santo Tomas University in Chile. So these are our panelists this afternoon, and I'm going to hand over. Our first speaker is Matthias. So if you would like to take over, Matthias.
Thank you, Katie. Hello, everybody. My name is Matthias Rillig. I'm a professor of ecology at Freie Universität, Berlin. Now, I've been working on mycorrhiza since my PhD, which started in 1993, which is more than 10 years ago now. And actually, then I already worked on arbuscular mycorrhiza, and I've been staying faithful to arbuscular mycorrhiza and avoiding other forms of mycorrhiza entirely. Also, since my PhD, I've already started working basically on the effects of various factors of global environmental change on the mycorrhizal symbiosis. And my lab now works on a whole range of topics. We do work on arbuscular mycorrhizal fungi still, and also how they respond to factors of global change, and also how they mediate plant responses to a variety of these factors. Now, when I think about threats, I'm sometimes asked what is the worst threat for soil, or in this case, what is the worst threat for mycorrhiza? And my answer in the last few years has always been the following. I don't think that there is necessarily only one single factor that is the single greatest concern, but I think it is rather the confluence of a lot of different factors that are all having their origin in human activities on this planet, collectively known as human-caused global environmental change, or for short, global change. And this is actually what we're currently working on a lot in the lab, and also more recently with avascular mycorrhizal fungi. So what I mean by that, by a lot of factors, is actually, first of all, more than two. And so what we know a lot about, generally speaking, for soils from experiments, not just mycorrhiza, is we know a lot about what one or two factors presented at the same time do to soil. And we are basically completely blind on what happens when more than two factors are active. So we did this also in a systematic mapping exercise, scanning all the literature, several thousand papers, and we found that 98.2% of all papers published on experimental work on soil have looked at just one or two factors. So we really don't really know what happens when more than two factors act. And so this is very difficult to study. We work with up to 10 and 12 factors simultaneously. This is actually not unusual. I think most people that do observational work in ecology would readily acknowledge that a fairly large set of factors can impinge on any one soil at any point in time. For example, also in an urban environment, like I'm currently in Berlin, it's very easy to come up with 10 different factors that would affect the soil. And by things like that, I mean nitrogen deposition from the atmosphere, elevated CO2, warming, drought, pesticides, microplastic, disturbances, PFAS, and a whole range of organic and inorganic chemical pollutants. So how we do that is we actually ask a simpler question about the number of factors rather than specific combinations, because the number of specific combinations, as you might imagine, explodes very, very quickly and becomes impossible to do experiments on. So we do a trick basically by randomly pulling factors from a pool of factors and combining them in sets that have in common that they're exposed to X factors. For example, 5 factors where every replicate of the treatment 5 factors would by chance basically receive a different complement of these 5 factors. And doing that, we have basically found out that there is a steady decline in diversity and in soil processes and functions. And we have more recently also done that, observed that in a field experiment here in Berlin with 10 factors. So the more factors act on plants and their mycorrhizal fungal communities in a grassland, the more basically we see a decline in AM fungal abundance. We haven't done the diversity yet. That is next. And so when you ask yourself, what should we sort of recommend as a conservation measure? I've been thinking about this for a bit now, and which is a bit weird, I'm a little bit hard pressed to come up with something specific for mycorrhiza, because mycorrhizal fungi, especially AM fungi, are very unique in their ecology, and you'd think you'd come up with something specific. But I think the picture that is emerging now in soil ecology is that it is maybe a little bit misleading to just focus on one particular group of organisms when it's rather the interaction of a larger suite of organisms that should be in focus. So I think the recommendation would be, of course, to take care of soil biodiversity as a whole. That includes mycorrhizal fungi. And basically, we have recently done a meta-analysis that showed, as additional ammunition for this argument, that showed that AM fungi basically improve plant responses to every factor of global change that we examined. This is elevated CO2, warming, drought, and nitrogen deposition, the big 4. So in every single case, looking at all the data published so far from experiments where mycorrhizal fungal presence and absence was modulated, we see that with mycorrhizal fungi, plants respond more favorably to that factor. So that basically is very good ammunition for saying that we should really take care of mycorrhizal fungi in soil if we want our plants to better deal with global change. And not just plant performance. We recently also did a greenhouse experiment where we, this is not published yet, could show that in the presence of mycorrhizal fungi, also the soil functions as a whole respond better to the impending changes. And that's it from my end. And back to you, Katie.
Thanks, Matthias. That's lovely. Great. Thank you very much. So our next speaker will be Nguyen. So I'm going to hand over to him.
Yeah, Katie, thank you so much. Let me get this started here. Okay. Okay, how does this look?
Perfect.
Okay, well, thank you. All right, so aloha everybody, and greetings from Hawaii. I'd like to maybe hone in on and following on Matthias' message a little bit more on these factors. And so my lab study and has continued to study the interactions between fungi and bacteria, and that's our main concept of study at the moment. And what we're looking at is the ecological outcome of these interactions and what effects they have on ecosystems. I have and continue to study mycorrhizal, in particularly ectomycorrhizal fungi, and their life history strategies, including systematics, and there's of this thing called Suillus, a very common and widespread species across the world. And currently, I am leading the International Suillus Consortium, which is a group that is trying to build a genus model model system for studying mycorrhizal fungi, or at least as a model for plant fungal mutualisms. And so we approach this from a genomics perspective. Right now, we have over 40 genomes across the whole genus, and many more of introduced species of Suillus across the world. And so I just want to say that if you study Suillus or you're interested in this group, join us. We have a consortium. You can send me an email, and we'll add you to the list. So let's dive into what I think of are threats to mycorrhizae. So let's pose this question: what happens to mycorrhizae if the trees disappear, or vice versa? Right. So in mycorrhizae, there is this concept of redundancy in that a certain tree can host over, say, 60 species of ectomycorrhizae. So a tree is not only dependent on one species. From a mycorrhizal perspective, from a fungal perspective, it can colonize perhaps multiple tree species, but not all, because some mycorrhizae are host-specific. And so here, if the trees were to completely disappear, I think the mycorrhizae will follow. Now, I'd be remiss if I don't mention Hawaii and its status of plants. As we know, these mycorrhizal plants or fungi are connected very tightly to plants. So here in Hawaii, we have 31% of our native plants are endangered, and that makes up about 44% of all the endangered plants in the US. A recent thesis by Jerry Coco out of Nicole Hinson's lab here showed that about 97% of native plants are associated with arbuscular mycorrhizal fungi. So that's a pretty high proportion. Now, we still don't yet have a clear idea on the species that are here. And so I think there's a lot more work to be done. But the bottom line here is that to conserve the host, to conserve the mycorrhizae, you need to conserve the host. There's perhaps an alternative strategy that we can take. And I'd like to propose this spore banking concept, in that similarly to seed banking, we can take the seeds of the plants and put them into a vault to save for a time when you need to revive them. Can we do this with mycorrhizal fungi? So this is following a long-term study. This is an image of my advisor, Tom Bruns, back in the days. And what he did was took spores of these rhizopogon mycorrhizal species and bury them in the field. And over time, follow the progress of their longevity. How long can these spores actually survive? The patterns after 15 years, this is a figure that summed up 15 years worth of work, shows some interesting patterns, but I won't talk about that right now. What we do get and what is more pertinent to this talk is that these rhizopogons are viable and up to 15 years. And so certain species can be stored this way, but not all species are receptive on seedlings. And so it's that strategies can work for some species, but not. And the last concept that I'd like to bring up is not as a threat to mycorrhizae is this concept of species concept in arbuscular mycorrhizal fungi, in that we don't really have a really good species concept. We do have pretty good species concept for ectomycorrhizae, but not arbuscular mycorrhizae. And so arbuscular mycorrhizae, I categorize them as these two concepts, the broad and narrow concepts. In broad, that if you take a broad species concept, that means that these species are everywhere, maybe a couple hundreds of species of mycorrhizal fungi across the world, arbuscular mycorrhizae across the world. And what this can lead to is that you missed ecologically adapted variants if you're interested in preserving certain types of fungi. Now, if you take a narrow approach, and currently this is sometimes what we do in more broad environmental sampling, we use the ITS gene, which can overestimate. And so we are now unable to prioritize which fungi to conserve. And so I think as a community, we need to define a species concept for arbuscular mycorrhizal fungi that hopefully can make use of current data, and this will propel us towards reducing the threats to these mycorrhizae. Thank you.
Great, thanks, Nu. That was great. So our third speaker this afternoon is César Marín. So I'm going to hand over to him. Are you okay, César?
Thank you. Yes. Do you see my screen?
Yes, we do.
Okay, great. Thank you, everyone. Thank you, Katie. My name is César Marín. I am a full professor in Santo Tomas University. I'm also editor-in-chief of the International Mycorrhizae Society newsletter. So if any of you have like articles or mycorrhizal research that you want to share, please contact me. Well, threats to mycorrhiza. This was quite something hard to think about it. Well, first, my mycorrhizal research, sorry, in general is related to fungal biodiversity, ecosystem functions, some stuff about applications in agriculture and restoration, and very recently the subject of multi-level selection, how mycorrhizal collectives that I define as the sum of the host plant plus the AMF fungi in particular and the bacteria living in the AMF hyphae can form like a evolutionary individual that is subject to natural selection. I do this research in several countries, mainly in South America, and it's a myriad of topics, as you can see in the slide. Applied work in, I don't know, restoration of conifers or in vineyards across Chile, but also application of AMF directly in situ. But also some base knowledge like effects of altitude, precipitation, environmental drivers of dual mycorrhizal plants, and some projects with the Society for the Protection of Underground Networks, SPON. So I think that the threats to mycorrhizae, they summarize in three major topics: in land use change, in conventional agriculture, and in global climate change in general. And this can disaggregate into many different topics or many different threats, like logging, urbanization, especially excessive fertilization, tillage, pesticides. In Argentina and in Chile also, we have a big issue with exotic fungal invasions, for example. But all of these are caused by these three major topics. So one of the questions that we received was how my research addressed these threats. Well, I think that in one side, one can promote like a sustainable agriculture framework that potentiates the mycorrhizas already present in the system, but also adding new ones. But here is an asterisk about adding new mycorrhizas because there is a lot that we still don't understand, although this research has many decades, but there is a lot that we still don't understand about the impact of adding new, especially non-native mycorrhizas to a system. So I think that we still need to do a bunch of studies in this. And I think honestly that a big threat is that we simply don't know enough the fungal biodiversity in many parts of the world. So one of the ways to address these threats is by filling these gaps in knowledge through initiatives like this, like GSBI and many different others, like the one that I lead, which is the South American Mycorrhizal Research Network, but many others. And here, for example, you can see in the left side an article by Alejo. There are some co-authors showing the endemicity of two major mycorrhizal types, arbuscular mycorrhiza, nectomycorrhizae, and you can see that in some parts of the Cerrado ecosystem in Brazil, for example, or here where I am right now in Patagonia, uh, the predicted endemicity of these both types is huge, is quite big. But on the right side, you see the literature that we scouted on these different mycorrhizal types, and you see all the gaps, all the white space, all the places that have not been researched. So I think this is still an important issue. Um, and finally, how can mycorrhizal research inform policy to protect mycorrhizae against threats? Well, this was the most difficult question, I think. But again, I reiterate that we need long-term in situ research to understand mycorrhizal applications. We, as a mycorrhizal researcher community, need to be very honest and not overhype and overselling mycorrhizas. We need to understand the complexity of the relationships. As Katie introduced, they are in a continuum between mutualism and parasitism. So they are not always good for the plants, and they depend on nutrients, they depend on other microorganisms like bacteria, and this complexity needs to inform policy. Again, we need to fill the gaps. We need base collection of data. We know just like 14,000, the mycorrhizal type of 14,000 species of plants. That's very little. As Matthias say, we need to study all these factors together and deeper study of interaction with other organisms. And that will be back to you.
Fantastic. Thanks, César. Okay, that's great. So now, if all of our speakers would like to turn their cameras and microphones on, we're going to enter into the discussion part of today's session, which is the best bit, in my opinion. So I'm going to open the question and answer. So just to reiterate, if you've got questions for our speakers on any aspect of anything they've talked about or something else entirely within reason, please type it in the question answers box. We won't be taking questions from the chat. Cool. Okay. So I'm going to kick it off with the first question from the Q&A box from Colin Averill. How can we quantify soil mycorrhizal and microbial biodiversity recovery in a restoration context? How do we know if soil biodiversity in a particular location is degraded or if a management intervention is facilitating recovery? Does anybody want to get started on that one?
Well, maybe I can give a sort of a first stab at that, and then somebody else can continue. But I think that is a question about the reference sites. And in some areas, reference sites are probably hard to come by. I would be hard pressed to say what a reference site here in Germany would be in reference to what. In the reference to the absence of human activity that in this densely populated part of Central Europe is impossible to come by. In fact, a little while back, I wrote a paper that there is basically no control anymore. Global change has engulfed, it's maybe too serious a word, but has affected literally every part of this planet, even parts where no human has ever been nearby. I mean, microplastic rains down from the atmosphere, just as an example, and warming affects the globe. Elevated CO2 concentrations are happening everywhere. It's well mixed in the atmosphere. So I guess the question is very tricky because a reference site with respect to what? And I think that is a central unsolved issue in my mind with restoration ecology. Basically, what are we restoring to? When do we decide it has been a success, given that we are on a trajectory of change that is basically impossible to stop and to reverse? I think you can maybe ameliorate some of the worst consequences of some drivers. Climate change, refugia for climate change, but then it ends. So I think that is an interesting question, and I think it is impossible to give a very good answer to it.
No, it's a good attempt though. It's an interesting point, and where are we restoring to, and should what we're restoring to be appropriate for the new conditions at that place in that time, right? You can't just go back to what it was, I guess. It's an interesting point.
And if you restore it to something and then this trajectory continues, it may just nullify all your efforts. Yeah.
Absolutely.
Maybe I'll add just something to that. There is a growing concept of soil health, and you can define it in many ways, but it to me it provides you a way to maybe not need this baseline, in that, okay, what are we defining as health for the current moment? And can we bring it to whatever you're restoring to that moment? And so how do we measure that? Right? We have tools. Some of these tools are still being developed. And molecular tools, I think, are going to be very important for this, because when it comes to soil, it just gets very messy. So we need powerful tools such as molecular tools to move us forward. And so if you can put molecular tools into the framework of soil health to measure restoration, maybe you can start pushing it to where you want to go. Now, I do realize that this is very messy, and we've been thinking about how to do some of this in our own group here in Hawaii. But it is something to work towards too, as we, like Matthias says, tackle the changes that we're dealing with.
Great. Thanks, Nu. So I'm going to move on to the next question, which I think, César, I think this kind of relates to the stuff you were talking about that you've been working on. It's a question from Laura Rillig. What is the role of AMF for sustainable agriculture? So it's a nice broad one for you. And then what are the practices? So I guess that practice is, how do we enhance AMF in agriculture? Do you want to start us off on that one?
Yes. Well, I think that as everything, it depends highly on the context. I would say that in sites where there is hope, in like an agricultural sites where you find some hope, by hope I mean at least some minimum amount of soil biodiversity and ecosystem functions, you need to implement practices that sustain what is already there. And this could be no tillage, would be a classical example. But in Chile, we have found places which have definitely almost no mycorrhizae, like 2 species maybe. And these 2 species maybe are not so mutualistic because they, I don't know, the phosphorus condition is 100 ppm or 150 ppm of also in phosphorus. So they are almost no mutualistic. In these kind of sites where definitely there has been centuries of conventional agriculture. I think that it's appropriate to introduce biofertilizers, but still following their ecology, following their effects, like making nice experiments, in situ experiments mainly. Most of the research in AMF is on greenhouses, but we need to measure like the real stuff to see the effect of these AMF biofertilizers. Typical practices that we have shown and many other people have shown that increase the AMF biodiversity already there. Could be crop rotation, could be polyculture, right? And organic farming practices in general. Yeah.
That's great. So just to follow up on that, this question led on to a few more questions in the chat, and I think we can kind of address those ones. So we've got one here. You mentioned it very briefly, César, but I wonder if you could just go into a little bit more detail for us. Recently published research, this is Naomi Zurcher, sorry, recently published research has shown that fertilizers can negatively impact mycorrhizae. Does this include what is classified as organic fertilizer?
Oh, that's a good question.
Yeah, it's quite a tough one, right?
Yes.
I'll throw it out to you guys, because I'd like to know.
No, to be honest, I don't know if it includes organic fertilizers. Yeah.
Right. What about Matthias or Nguyen? Do you know these impacts, the negative impacts on mycorrhizas of, I guess, just high nutrient environments, right?
Yeah, well, it's a high nutrient environment, and that is a condition in which some of the services of AM fungi are not in great need, and where the plant will start regulating down the symbiosis. And if that happens on an agricultural field, you have high nutrient conditions. You have right off the bat, you have a non-responsive host plant because it's been mostly bred like that. You have the occurrence of fungicides. You have tillage, as César has mentioned. So I think that there is, nutrients is but one of the very many factors that mycorrhizal fungi have to contend with in an agricultural context. And certainly high nutrient environment would be deleterious. Depends also on what nutrient, eh? I mean, if you have high N, it can actually kick in the mycorrhiza because the plant is getting more starved for P. With organic fertilizers, I should also add that what you are adding is not just, of course, nutrients. Especially here, you are also adding plastic. This has been quantified and is unavoidable. You're also adding antibiotic resistance genes, depending on what the source of the organic fertilizer is. So this is a rather complex question to answer satisfactorily, but it is more than just nutrients would be my addition to what César has already said.
Yeah, and I wanted to add that there is a very nice recent article in Nature Microbiology by Marcel van der Heijden showing kind of the predictors of inoculation success. And if I remember correctly, one of the main predictors was the pathogenic fungi in the soil microbiome. So I think that under some context of high abundance of these pathogenic fungi, yes, the inoculation can be a better measurement. But in the same article, they report like a high percentage. I don't remember if it was 25 or 40%, in which the inoculation actually was not successful or had detrimental effects. So again, we always need to be very careful and check all this through time and with many different factors. Yeah.
That leads me
Maybe if I can add to that with this inoculation.
This was
Yeah, so sorry, I mean, the inoculation always elicits an allergic response on my end because, if you change nothing else in that system and you're just adding AMF inoculum, maybe you have some short-term effects and you're just dooming those poor things to die. Because if you have changed nothing else, then I'm not sure what you're gaining by just adding AMF. I think César has already said that if you are into restorative and regenerative agriculture and you're having nothing left in that soil to do that with, then maybe you do some locally sourced AM fungal inoculation. But other than that, if you don't change anything else in that system, that seems to be completely futile. Yeah.
Yeah, I mean, this is partially answered the next question, actually, so it's a really good point. I'm just going to bring it up and open it up to all three of you, because I think it's a really interesting one. It's a really important question as well. What do the speakers think about all of the mycorrhizal inoculum available commercially? Is this a threat to more locally adapted or host-specific mycorrhizal associations? So I think, Nguyen, you talked about specificity and associations. Do you want to start off? What do you think about commercial inoculation or inoculation?
I'm stab myself here. So my feelings on those is you got to be very careful before you buy. How old those spores have been sitting around. As far as I can reach back into the literature, there hasn't been any work done on longevity of arbuscular mycorrhizal inocula. As far as I can reach back, it's about 6 months. They probably can go for a year if you store it right, but you have to think about how long these spores actually last when on the shelf. That's the first thing. So are you buying dead spores, right? So we need more research on that area. And in terms of specificity, most of the companies that make these inocula, they will take a single organism like Rhizophagus, and one thing and try to pick an inoculum that's going to work for broad types of plants. So when that happens, you go to an environment that is not where those fungi came from. They might not work very well, and they might not be able to compete with the native fungi.
Yeah, agree.
Yes, just to add to that, there is a nice study by Matthias Salomon, 2022, I think, where they tested like 25 of these products in two different continents and found basically that 80% of them don't work. So that's very important to consider. By don't work, I mean they don't increase plant growth, basically. And there is also another one by Tanel Bater and the team in Tartu, sequencing and showing that also in the majority, the uh, declared content, the declared content of a species that doesn't coincide with what they found by sequencing. So I think this is what I meant by too much hype in my presentation.
So this is a beautiful segue into our next question then. So Nancy Johnson's put a question in saying that she appreciates César's goal of less hype, more hyphy. Do you want to just elaborate a little bit on that recommendation and provide some recommendations for accomplishing that? So it'd be good to hear from each of you what you think we could do to, decrease hype and increase hyphy. I'll start with César because you started that part of the conversation.
Okay, yeah, well, I think a big part is what we are doing precisely at this moment to have these honest and open discussions with the general public to present the complexity of mycorrhizal interactions, to show, for example, Katie's work on endophytic fungi, right? That there is plenty that we still need to know. I think that yes, because we all do the presentation when we show that there's a positive interaction, and just it seems to me that sometimes that's the main message that people get, that the plants give something, the fungi give something, but it's a little more complex than that, and we should present this complexity, the importance of environment. We need to show that many times they don't work, basically, for agricultural or restoration purposes. So I think that's a more open and honest discussion with the general public. Yeah.
Thank you. And then Matthias, have you got any thoughts on this?
Maybe just that it is rather symptomatic, I guess, of our sort of society that we seek technological fixes to things that have been caused by technology in the end, like industrial agriculture, which of course feeds us. But yeah, so that maybe just to say that there is never going to be this magic bullet that solves it all. I think that you have to really affect a system change towards more regenerative practices overall. That includes many of the things that have already been mentioned here: crop rotations, don't have fallows, maybe avoid plowing unless necessary, reduced use of chemical compounds like pesticides, and a responsible use of fertilizers. I mean, you have to add fertilizer because you're harvesting things. So I think rather than this magic bullet stuff that always leads to the hype, this one thing that's gonna save us, maybe engage in the admittedly much more tedious and complicated discussion that this is gonna change, many parameters in the system have to change. Yeah.
And then, No, if you've got any points to add on to that.
Not much, I think. I think César and Matthias has already covered the points, and I agree with them. Definitely, I think we need to. Okay, well, I'm very excited that the public is excited about fungi. That it's for a long time we've been mold under the cupboards, right? But now it's in the public's eye, and people are looking towards fungi for solutions, which is wonderful for the field. But the communication between the science and the science the public needs to be better tuned, and I think that is one important aspect for us.
Yeah, I think that's a really important point. I think actually also, Matthias, go on.
Oh, sorry, I didn't want to butt in. Just wanted to say that what Newt just said has triggered something. We had this discussion in a lab meeting actually on Monday. We discussed this paper in Trends in Plant Science, where the books by Wohlleben and Simard were sort of dissected with respect to the veracity and the to what degree are these statements in these books really supported by science. And in the beginning, I was actually of the opinion that the end justify the means. I mean, if people are excited about nature and trees and mycorrhizal fungi, that's good. Even if these books deviate from fact in the beginning. By the end of the discussion, I think I've come around to the personal conclusion that people can, of course, disagree with, that maybe there are short-term gains and net benefits of books like that, where unsupported facts are basically used to get people excited about fungi, mycorrhizal fungi specifically. But I think the long term of such science communication is going to be bad, because when, as it just happened with the paper by Justine Karst, and we also have some coming out, that when it shows that, or when it's being demonstrated that these things are not so, that these are actually not true, then it may actually cause a backlash in the public by saying, oh, well, this has all been basically untrue and unsupported, or they have lied to us. And so maybe then we don't want to hear this anymore. So I don't know if I can phrase it in the right terms, but I have really flipped my opinion on Monday, basically from it's fine, don't worry about it, to, oh no, this actually has probably long-term consequences that are actually quite bad. And actually, what these books also don't do is portray science as a process. They say things are just so, use anthropomorphisms for things that really don't need to be anthropomorphized because they're excellent and interesting in their own right as organisms. And yeah, anyway, so I think that I wanted to say that too.
And actually, it's kind of a bit of a personal anecdote. So I was talking to someone who I know works at Kew, Royal Botanic Gardens at Kew, and they've been taking members of the public out on fungal forays and walks, and they'll be talking about mycorrhizas. And they got asked, oh, the Wood Wide Web, right? But that's not true, is it? So do mycorrhizas not exist? So, you know, it can be really damaging and undoes the good that has been done. But equally, there is a role, and it's kind of difficult to strike that balance. There is a role for getting people excited in the field and bringing attention to the field, right? So it's that point about communication and accurate communication and letting people get excited about stuff that's actually true. I think is key. Yeah, really interesting discussion. So I'm going to just change topics slightly. So we had a couple of questions come up about something new that you brought up, and it was about this species concept for AM fungi, and I wrote it down as well, and I thought it was a really interesting point that you made, and I wonder if you wanted to just talk a little bit more about the challenge of developing the species concept for AM fungi. I think what we're being asked, I think both of these questions that I'm looking at are kind of asking a similar thing. What are the barriers to defining a species concept? Why don't we have one for AM fungi? Why is it so hard? Can you just explain a bit more about that, please?
Sure, yes. So for a long time, AM fungi has been defined by morphological characteristics, which in non-mushroom making fungi, it's relatively difficult, right? There's not many characters you can go on. And so spore morphology was used mostly for this. And with spores, you can have two things that are quite different, looking relatively similar. And so that gives us a couple of hundreds of species of fungi based on this concept. So when we started sequencing DNA, we found out, hey, there's actually a bit more using the large subunit ribosomal RNA, which is the standard in the field. Now, when we started doing community ecology, we found that, well, you can own the marker genes that we use is the internal transcribed spacer, the ITS gene, which overestimates the amount of diversity within a single arbuscular mycorrhizal fungus. And so therein lies the challenge. How do we bridge the morphological concept, if you believe in that, to the more conservative large subunit ribosome concept, to the functional or the what we do in community ecology, the ITS concept. So each of these gives you a different answer, right? So that's where we have to connect to build a more useful concept. And it's hard because we all do different things, and how you apply it is going to be different.
Yeah. Have you got any suggestions of how it might be achieved?
Katie, that's a very difficult question.
Ready.
I think there's definitely been published paper going back and forth about what's a better way to do this. And I think there've been some consensus, but not really. So I think the people who are passionate about this has to get together. They have to get together in a room and have a real discussion. Probably a heated discussion, but they do need to have a real discussion. So we can decide some ways to make this concept viable.
Yeah. I mean, maybe a meeting in Manchester in August would be a great place for them to do that. Maybe a meeting on mycorrhizas, just okay. Right. So let's talk a bit more. Let's switch to restoration. So we touched on that earlier, and we've had a few more questions come in about restoration since then. And I'm looking at a question here about oh, I've lost it. Oh dear. I did have a good question. Okay, here it is. So how should ecological restoration goals evolve in the Anthropocene era beyond the conventional goal of restoring ecosystems to their original state? Specifically, in what ways can we harness microbial communities to bolster resilience and facilitate the growth of ecosystems that can adeptly respond to shifting environmental conditions? I don't know, who wants to take that one on to begin with? César, we've not heard from you in a while.
Well, that's a very difficult question. I think that, yes, as Matthias said before, it's very difficult nowadays to define a base ecosystem. I think this is no longer the case in most recent research, but maybe to take an approach on ecosystem multifunctionality and to see the evolution or the change of these restored places in regards to as many soil functions as you can measure would be a nice approach to see if there is an actual increase in this multifunctionality. And how can we harness microbial communities? I think there is starting to be a consensus, or not a consensus, but still a growing part of research regarding artificial selection. So you would have like a whole soil microbiome under different treatments, or with the same genetic basis of the same plant species, right? The same phenotype. And you measure first in pots the plant response or the ecosystem function that is your focus. And then you subtract that microbiome for the following generations. This is still kind of very new research, although there is a nice paper from 2000 in PNAs that already they did this, and it shows like a linear response to I think they were selecting, yeah, for plant growth basically. So it shows like a linear response under different generations. So I think this is an approach that needs more research. Well, of course, because soil is a finite resource, so it's not like you are going to extract soil all the time. So yeah.
Great. And then any other comments from our other panelists on this subject?
Yeah, so I mean, what César was describing, I think, is microbiome engineering, and that is something that is often pops up in that context, that you may be able to generate tailor-made microbial communities that you can use to supplement the existing community in a soil. What all of these papers that propose that idea forget is that there is not going to be a sterile soil to which these microbiomes, the engineered microbiomes, are going to be added, but they're going to be resident communities, and then you have what we call microbial community coalescence, so the encounter of entire microbial communities with very uncertain outcomes. So I think that there is a lot of research that is still needed in order to make that happen. I mean, there are very nice papers that have recently come out with, inoculating soil for more drought area and whatever that helps soils under more drought conditions. That is sort of something like that, or soil transplants. But there is a lot of uncertainty in the addition of that microbiome engineered community to the target soils.
Great. And then Nu, have you got anything to add on to that?
It's just very briefly, just following what César was talking about. I really do think that conservation or restoration efforts should try to bring an ecosystem to its functional maximum, however you define that. And so when you think of a degraded ecosystem, its ecological functions are no longer there, right? So a forest is no longer behaving as a forest. And so instead of trying to bring back the same species, or the type of thing that has been there before, the changes around the globe is going to make that difficult. And so can you bring this ecosystem back to its functional maximum, as you think a forest should behave, would be a more important goal. At least for me, I think it would be an important goal. If you're getting the nutrients to flow correctly, you're getting the gas exchange correctly, and these are the functions that we want. So it's a hard goal.
Right, which leads us nicely into the next question. This is kind of still on that engineering the microbiome and applying mycorrhizas in an applied context. So the question is, do you feel we are confident enough in our understanding of mycorrhizal associations to perform translocations more regularly as part of agriculture, forestry, or conservation interventions? Obviously, we wish to avoid non-native species, but we know native mycorrhizas are an important part of plant adaptation to a changing world. So should it be a priority? So we're kind of moving into that changing climate and mycorrhizal sphere. Give you a second to think about that, because it's an interesting thought. What do we think? Do we know enough to be able to be transplanting native mycorrhizas around? Anyone want to go first?
Well, I think what is the species that you're transplanting? And so it goes back to our need to define the species concept, right? If you're going Rhizophagus into radices, okay, that thing is maybe it's everywhere, maybe not. So you got to define what it is first.
I think as a personal anecdote, one colleague here in Chile, she has been very successful in applying consortia of AMF, like 5 species from 4 different genera, obtained from soils with high aluminium and very low phosphorus. When these consortia are applied in similar soils, they are very successful in general. When the soils are quite different or is way northern side, not so much, although still good results, but not so much. So I think that we might know enough, but this knowledge needs to be very specific, needs to be region specific or soil specific at least.
Excellent, thank you. Matthias, have you got any?
Yeah, maybe just as one additional point, just to reiterate, I think that should always be the last resort. You should be trying to manage any residual populations that are still there by changing management practices so to be more conducive to AM fungal biodiversity rather than bringing them in from the outside, because that means you're going to be mining and destroying soil someplace else inevitably. It's got to be can hardly be sustainable, I think, at the levels where you will need that. So I think that should be the last resort, and the focus should be on managing what is left.
Okay, so let's stick with soil as our focus for this next question. So should our starting point then be understanding soil structure and function? So that's compaction, porosity, drainage, macropore status, variability, and the relationship/association between upper and lower horizons, i.e. the soil basis for organic biodiversity. So do we think like managing and understanding those factors should be the starting point for where we start to apply mycorrhizas? Or are they, I don't know, have you got other thoughts around that? Please jump in if you have a thought. Yeah, go for it.
Maybe one thought to start is that it depends on your goal. So, I mean, if your goal is more to favor plant growth or yield or improve any yield component, yield quality or something, then maybe you would be looking at favoring different processes than soil structure. Since soil structure is my personal hobby, I think that I'm always in favor of that, right? And because it has a bunch of consequences, but that are positive for carbon storage and so on and so forth. But I think it depends very much on what you have defined as your goal, what you should be focusing your research attention on.
What about our other panelists? Have you guys got any thoughts? No? Okay. No? Great. No, I think you covered it. Thanks, Matthias. So let's stay on that sort of theme of global change and mycorrhizas then. So in regards to addressing climate change and carbon dioxide drawdown, can any of the panelists address the role of AMF in stabilizing root carbon exudates, I think we'll include ectomycorrhizas in this as well. So just any mycorrhizas in stabilizing root carbon exudates in mineral-associated organic carbon. Is the mycorrhiza also weathering minerals that are associated with this root carbon? I'm getting vigorous nodding on the right side of my screen. So I'm going to hand Nu. Do you want to explain your nods?
So I think the question is about drawdown, right? And the consequences of putting carbon into soils as well as removing carbon from soils by fungal activity. And I think that's a very timely question as the soil science is moving at the moment. We know that certain soils can hold a tremendous amount of carbon, but the latest research shows that there's a maximum of how much carbon a soil, a mineral surface can hold. So you can replace that, but it's just going to be a maximum. And so that's going to be a challenge for us to figure this out. Certainly, the activity of the mycorrhizas, they say organic acids, can liberate large amounts of nutrients like phosphorus, for instance. So, yeah, I think we need to find that balance.
Matthias?
So I think to jump in here, some people disagree that there is a maximum because it's not just determined by mineral surfaces, because there's also organic matter interactions, and mineral surfaces wouldn't limit them. But yeah, I'm not in a good position to argue for one or the other, just saying that there's a bit of uncertainty there, and it's the jury is definitely out on if this is true or not. And there's also evidence that suggests it just can go on. Probably not in every soil, yeah. Yeah, I mean, we recently have a review paper on mechanisms of carbon stabilization via AM fungi, and so there is also AM fungi can be involved in mineral weathering, since that was, I think, a component of that question. And of course, one way is, that the hyphosphere, so the bit of soil around hyphae of avascular mycorrhizal fungi, can feed certain bacteria that can have an important role in soil carbon sequestration or processing, more in general terms. And then finally, the physical and biochemical effects of the hyphae as a whole can, in conjunction with other soil organisms, produce aggregates. And inside of these aggregates, and the mineral surfaces inside of the aggregates, carbon that would otherwise be labile can be at least temporarily stabilized. And we do know that the chemical structure does not necessarily predict persistence. I mean, this is a relatively new paradigm, so that, it's the location, it's where it's at. And so I think AM fungi can be very important in that, also in their own necromass production, where they sequester that carbon. I mean, they can reach small nooks and crannies and crevices in the soil pores that are very difficult to access by other microbes that could process that carbon. So yeah, I mean, there's a variety of different pathways that are partially well-researched, partially relatively new, via which AM fungi can contribute to the stabilization of carbon once it's in the soil.
Okay, great. Thank you. Great. So I've got here, there's a question for César. You mentioned that more in vitro studies should be carried out to better understand the mechanism, effects of different elements of climate change and artificial change to mycorrhizas and soil microbial communities before testing in the fields. So it says, I totally agree. I am currently working on AMF in vitro cultures. However, most of the in vitro lines are rhizophagus, and I'm always wondering if this can really represent the AMs in the soil. Do you think all these studies on rhizophagus and concentration on this one genera are leading us to a biased direction? What do you think from an ecological point of view? I think this is a question for all of our panelists, actually, but I think, César, if you can go first, that'd be great.
Yeah, that's a good question. Well, actually, I mentioned that more in situ studies are necessary, but definitely the selection of a few species of AMF has a lot of bias. There is a nice paper by Nicolas Marro, 2022 in Neophytologist, showing the different effects of different AMF taxa. So some are more related to phosphorus nutrition, some are more related to plant defense, some taxa. And I did emphasize in my presentation as well the study of these non-nutritional effects, which I think they are quite important or as important as the nutritional effects or the resistance to abiotic and ambiotic threats, let's say. So yes, I confirm or affirm this question. I think that this introduces a lot of bias and definitely more basal taxa and more recent taxa in AMF at least should be contrasted, or taxa that prefers to live inside the roots and taxa that prefers to produce more extra radical mycelium should be at least contrasted in this kind of studies. Yeah, definitely.
Thank you. And do our other panelists have any thoughts on this? I think it was a pretty comprehensive answer there. Thank you. So I'm going to ask a few questions now from the box that focus on methods, because I think people are asking for like best ways of doing stuff, and it kind of leads on from we should be using other species of fungi. And one of them was, where is it? I've lost it now. Here we go. So what are your tips? So everyone's done work in mesocosms before, but what are your top tips, panel, for manipulating plant mycorrhizal interactions in a mesocosm? This is the first of a couple that are going to be methods-based. So I want to hear from all of you. What are your best tips for these? We'll start with Matthias, because I know you do an awful lot of mesocosm work.
Yeah. So I mean, yeah, I don't know what the best tip is. There is, as you all know, no specific way to eliminate AMF from a soil. So the approach is to eliminate everything and then selectively add AM fungi to one series of your experiment while not adding it to the other. In doing so, I think it's very important to emphasize that all experimental units have to be steamed or autoclaved or gamma irradiated or whatever you do. Otherwise, it becomes completely invalid, because then you just have basically changed the soil physicochemical conditions in just one part of your treatment. So it's important when you do that, basically starting from a clean slate, building this back up, that you treat all of your samples that way. And then you add, by virtue of their size, you could add mycorrhizal fungi, either isolated from a site, or you could buy them from a commercial supplier or whatever you want. And then it's important to not forget that the control in mycorrhizal addition experiments is always triple the amount of work as the addition of the treatment, because you need to control for all the stuff you're adding, you know, so you need to add autoclaved inoculum to the control to equilibrate the nutrients and a little bit of substrate of some random stuff that you added in your experimental units. And you need to do a microbial wash from your live substrate and add that as well to your control. So, you know, that is a very nice illustration of the fact that a control does not mean do nothing. In fact, in this case, it means do 3 times the work. And then, you know, then you equilibrate the soil microbial community by adding microbes from a microbial wash, so you get the microbial community to reestablish in these autoclaved or steamed or whatever have you experimental units. So it's challenging. What we also use is the famous Dave Johnson rotated, non-rotated core arrays, but there you basically create a soil that's under the influence of AM fungal hyphae or not. That works reasonably well, actually. We've also tested for non-target unintended effects of this rotation. Most people don't do that, but we have done this now several times, and we find none of these effects, which is nice. Yeah, and yeah, I think that's the main points from my end.
I think those controls, you know, there's so many papers or manuscripts that I see that don't have the controls. It's actually an experiment of testing inoculum versus nothing, and you're kind of like, well, yeah.
Yeah, it's not completely wrong, but you're asking a question that is different from the question you thought you were asking. You're asking about adding this inoculum with all the stuff in it, and so it's not the question you were actually interested in.
Yeah, exactly. And then you've got other stuff to add about working with mesocosms. Any top tips or hints for success? Or did
I've only
Matthias
done a
cover them
little
all?
bit of, sorry, Katie. I've only done a little bit of microcosm work, but it seems like you have to define what makes your plant relatively happy. Because when you see a plant growing in a field and you take that chunk of soil and you put it into a microcosms, the plants don't behave the same. There's going to be root competition, there's tightness in there, nutrients aren't flowing as well from other parts of the soil. So you're actually limiting what the plants are getting. So you might actually be changing the way that plant and fungi are interacting just by the fact of moving them into a microcosm. So that's definitely something to be aware of.
Such a good point. I have I say to everyone in my lab, and if there's anyone logged on, they'll be sick of hearing this. My question is, does your plant look happy? Because if your plant looks happy, you're in with a good chance of getting a result. César, have you got any additional points you want to make about my mesocosm work?
No, I think Matthias' answer was very comprehensive. I am curious about your answer, Katie, about this question.
What do I think makes a good
Yeah.
mesocosm experiment? Control, control, control.
Okay.
And also, yeah, make sure your plants are happy. Because if you've got a miserable plant, it's not going to be doing what you want it to be doing anyway. So you might as well start again. And I think adapting your question so that it can be answered with a mesocosm is important. I think if you try and ask too big a question that you simply can't get at in that system is a mistake that people make. I think that can be quite important. Oh, look, Matthias is desperate. He's jiggling around. What would you like to say, Matthias?
Me. And actually, when New was talking, I had this other thing that popped right into my head, and that is the temperature. I think when you were talking, that is exactly what I think is very often a problem, but rarely documented. Like, I give you an example. We have a very nice air-conditioned greenhouse. It's at 20 degrees Celsius. It has all the fancy shading and really air-conditioned and looks like a building wrapped by Cristo. It's got all these like shades coming down. It's fantastic. Air temperature really is 20 degrees or 19 degrees. When you measure the temperature in your pot, and I emphasize, in white pots, most people use black pots. The temperature in the white pot, in a greenhouse where the air temperature was 20 degrees, was over 35, going sometimes to almost 40 degrees.
Really?
And so I think if I were to build a greenhouse again, I would ask for one that has a root cooling compartment. But I think just as a general tip, worry about your root temperature. I have a feeling this is the main reason for failure because it simply doesn't get that hot out there. And your pots tend to be black. When we are forced by size and whatever to use black pots, we wrap them with aluminum foil. And we try to do everything to keep that thing as cool as possible. I am actually now convinced, now that we routinely measure the temperature in the pots, that this is a main reason for why things fail, just to add.
Yeah, you can't buy tips like that. Thank you.
I'll add one more, just because this is
go.
So I would say, think about the size of the pot. If you have a larger microcosm, it's going to buffer a lot of the changes. If you have a tiny pot, that's going to change very quick and heat up very quickly. Now, larger microcosms are more difficult to work with. So that's definitely something to consider as well.
Yeah, it's so important. So important. And you know, that's species dependent as well. Like one pot doesn't fit all, does it? You can't. Yeah, there's a lot. Okay, so staying on the technical side of things, top tips. I've got one about quantification of AMF. But this could also apply to ectomycorrhizas, and it's something I really struggle with as well. What would you all say is the best available tool to estimate the abundance of mycorrhizal fungi? It says in soils here, but I'd also be interested to hear about what you think about in roots as well. So yeah, I'm going to hand it over to you guys. Give me your thoughts or your top tips.
Well, at least for AMF, I think that we should still like teach taxonomy and despite what you said about the difficulties to define species just on morphology, but I very much value spore studies in AMF. I think they are very valuable and they can complement the metabarcoding approaches. And I know it's very difficult, it's very time consuming. Very few people in the world still do this, but I should emphasize the importance of doing spore analysis. Yeah.
Great. And that's just with microscopy?
Yes.
Great.
I would go molecularly in terms of I think the best way to get this is going to be PLFA, fatty acids. It gets you everything, including the mycorrhizas, but fewer and fewer labs are providing this service, so it's getting more difficult to do it. But quantifying microbes in soil seems to be one of those challenging problems that we can't quite get past. So currently, I think PLFA would be an approachable way to do it. You could also try qPCR maybe. That could also work. They don't give you the same answers though. They track, but they don't give you the same answers.
Everything gives you a different answer. I would politely disagree with the PLFA. I think it's quite well known that there's cross-reactivity with some bacteria. I think NLFA works and PLFA works inside of roots. Actually, what I always advocate for is do root colonization. Root colonization alone is per se not so interesting, but if you also have root length, you can calculate colonized root length. That is actually a parameter that can be directly related to a volume of soil, whereas percent colonization is dimensionless. And once you have colonized root length and you do hyphal length extractions, which are a pain in the neck and labor intensive and a little bit magic, what you count as an AM fungal hypha, because of the absence of specific stains. But then you have a really complete morphological set. When you have spores as well, it's even better. But I think as long as you have colonized root length and hyphal length, you get a pretty good sense of what's going on.
I'm scared of those hyphal length, Matthias. It is like you said, it's magic because there's a bunch of fungi that look just like AMF that are not AMF, and they're very common. Martyria, let's say for instance, no septa, they look just like an AMF hypha. They're common, and so it's very easy to mistaken them for AMF where they're not.
Yeah. You have similar issues also with other methods, so that's not unique to that. I do agree, and I have actually encountered soils where I could not do this because I saw trait combinations that made it impossible to make that call. So it doesn't work everywhere. I think if you work in a soil for like a couple of decades, maybe you're just consistently wrong. Yeah, so I agree. It's not fantastic as far as methods go.
Yeah. I mean, maybe there's a combination of methods. You can kind of, trouble is none of them agree. I think it was New said they don't ever match up. So it's hard. It's hard. Okay, but we've got some top tips there. That's great. Okay, and the final one, I'm interested to hear what your thoughts are on how can the exchange between plants and fungi be quantified? Who's got ideas on that? Anyone want to?
You should answer that, Katie.
I can answer that, but I'm not on the panel. I'd like to give you the opportunity. With my approach, I use isotope traces. So we use radioactive and stable isotopes to introduce to the fungus, and then we can track it back into the plant and measure how much of that compound has reached, or how much of that isotope has reached the plant tissue, and vice versa. So we can label carbon dioxide either with 14C or 13C, and then measure it, measure how much of it ends up in the fungal compartment in our microcosm setup. That does require quite specialized experimental design. So it's not applicable everywhere, and you can't answer all of the questions, especially in the field. It's very difficult. I think Dave Johnson does some really elegant stuff with stable isotope traces in the field with different mesh bags and things. So it's really about experimental design for that. But I mean, there are other ways. I know there's molecular ways of doing it. Does anyone want to speak on other ways of measuring stuff? No? Okay, well, I'll keep talking then. This isn't really, I didn't want to talk for 5 minutes.
No other ways, but I'd like
no other ways.
to follow up with your approach, which is the isotope approach. We use stable isotopes, no radioactive here, but stable isotopes, and the magic in that is we're interested in say carbon flow, right? Where does the carbon go from CO2 through the plant, through the fungi, through the microbial communities to mineral surfaces? So you can use stable isotope to trace the whole path of where everything goes and hopefully quantify it to a certain extent. So you can measure or rather you can quantify the amount of carbon that moves through the system. And so I have a project going on right now that does that.
Awesome. Awesome. So I think, yeah, that answers that question. So we've only got a couple more minutes left. So I've only got time to ask a couple more questions, and you'll see from the box, we have 24 lined up. So, I mean, we've had a lot of questions come in. So I'm really sorry if I didn't get a chance to ask the question that you've posted. And I'm just scanning through. I think we've got time for one more. Oh, I don't like being the chair. You got to pick questions. It's hard. Oh, sorry. I'm failing at the last minute. I'm really sorry. Okay. So this is just going back to okay, this is a future-looking question. We're going back to kind of our applied approach with mycorrhizal fungi, and I think this is kind of a nice one to finish on because, like I said, it's kind of a future-looking question. So someone has asked, mycorrhizas are a way to build resilience in crops and trees. However, they're also prone to global climate change threats. How do you see the possibility of use of mycorrhizas as a biofertilizer? I'm going to expand that to and other functionalities in the changing climatic situation. So what do each of you see as the future for applying mycorrhizas in a changing climate? Shall we start with Matthias on this one?
I can just quickly reiterate what I've already said, is that we showed in a meta-analysis that at least AM fungi really benefit plants with the big four: elevated CO2, warming, drought, and deposition. So it makes it better under every one of these circumstances for a plant on average over a wide range of different ecosystem types, plants, and soils. So it seems like there is very good potential for leveraging mycorrhizal fungi to improve plant performance under conditions of global change. That's the first part of the answer. The second part of the answer, you should do that by managing AM fungi that are already in the soil rather than by applying them if at all possible. And inoculation should probably be a last resort.
Great, thank you. César.
Yeah, I think that I partially agree with Matthias. Definitely management should be the first step, but also I think that we should identify areas of the world either for restoration or for agriculture which are like too far gone, like they have no hope. And in these places try to produce native consortia and native microbial communities, because it's not just mycorrhizas, but all the surrounding bacteria and archaea and so on.
Yeah, that's really important. It's not just mycorrhizas. Okay, and then new final thoughts on that topic.
Yeah, no, I would just agree with what Matthias and César has just said as well. Management would be the way forward.
Excellent. Great. Well, I'm going to draw a line under that then. Thank you all very, very much. It's been a really fascinating discussion. Thank you everyone online for joining in and posting all of your brilliant questions. And again, I'm sorry I've not managed to get through every single one of them, but hopefully I've covered the main themes that have come up in that chat. So I think it's Diana's just appeared, so I'm going to hand over to her.
I just appeared to thank you again. You guys did. This was really a wonderful, informative discussion, and I appreciate so much your time. And Katie, for your great job of moderating it. It was really a fun webinar. very informative. Thank you, guys. Thanks, everybody who came.
Thanks,
Lena,
everyone.
thank you.
Thank you. Thank you.
Bye-bye.
Bye-bye.